Hello, we are Christian Lundy, Ali Koehl, and Trevor Hile. We're undergrad students at Hope College, and we'll be presenting on research we conducted this summer studying the decomposition of peat moss. Globally, peatlands store twice as much carbon as is in the atmosphere as carbon dioxide. As average global temperatures continue to rise, these carbon stores will play an important role in the continuously increasing levels of carbon dioxide in the atmosphere. To better understand how carbon is sequestered in peatland bogs, we must further understand the Sphagnum genus of moss, also known as peat moss, which is the dominant plant in peatlands globally. Studies have shown that the high carbon sequestration in peatlands is due to low decomposition rates of sphagnum in a bog environment. Our study is investigating the mechanisms for this low rate of decomposition, specifically how differences in cell wall chemistry among different mosses affect the rate of decomposition. Our sphagnum moss was collected from the Minor Lake Bog in Allegan Township, Michigan. The bog has a microtopography of hummocks and hollows, each with different dominant species of sphagnum. 3 sample sites were chosen for each species. We also sampled 1 terrestrial polytrichome species to compare the decomposition rates to a non-sphagnum moss. Once back in the lab, the moss is clean of all extra organic matter, such as sticks, leaves, and insects. 15 incubation models were made for each sample site, each containing moss at 70% of its water holding capacity, and a native microbial inoculum. The incubation bottles were covered in foil to prevent further photosynthesis, and then stored to begin decomposition. For each of the mosses, we measured pH, iron content, and carbon-nitrogen content using an elemental analyzer. The incubation bottles were capped to trap carbon dioxide gas during decomposition, and the moss samples were analyzed by gas chromatography to measure the rate of carbon dioxide production.
We measured the buildup of CO2 over 24 hours and used that to calculate the average and standard error of CO2 production. Our data shows the rate of decomposition of the hollow moss is significantly higher than the hummock and the non-sphagnum moss. The control had no moss and was treated with inoculum but showed no CO2 production. These results supported our hypothesis that mosses from different microtopographies differ in rate of decomposition. The literature states that hummock mosses have higher concentrations of recalcitrant structural carbohydrates in their cell walls, and this is consistent with our observation of slower decomposition rates in hummock mosses compared to hollow mosses.
Our current work is analyzing these structural carbohydrates. Using our method, we've been able to make steps towards quantifying concentration of carbohydrates to determine whether or not they're defining factors for differences in decomposition rates. The method we have developed for analyzing dried ground-up samples for GC-MS include a methanolysis step and a silation step. The methanolysis pulls the sugars out of these samples as monomers, and we are able to isolate them and remove them from the rest of the sample. The silation step derivatizes these monomers so that they are detectable by the GC-MS. Our extended high-temp GC method then gives us beautiful peaks with great separation, as you can see here. Once we're able to compile more data from the moss samples themselves, then we'll be able to make conclusions on how these carbohydrates are affecting the decomposition rates between the areas. This will help us predict how peatland carbon cycling will affect climate change.